Pancreatic cancer is without a doubt one of the toughest fights in modern medicine. For decades, it's been this silent, fast-moving killer with very few good options. But what if we could actually read the enemy's playbook? I mean, what if we could see its secret communications and figure out a way to intercept them? Well, today, we're going to dive into the science of how a field called proteomics is giving researchers a brand new way to do exactly that. Let's just start with a stark reality here. Once a patient is diagnosed with pancreatic cancer, the median survival time is less than 6 months. That number, it really sets the stage for why a new approach isn't just important, it is desperately needed.
All right, so what is it that makes this disease just so incredibly difficult to beat? Let's break down the key challenges that make this cancer such a formidable opponent.
What's really fascinating here is how all these points connect. You see, this cancer is so lethal because it's an absolute master of defense. It literally builds this dense fortress around itself, what scientists call the tumor microenvironment, or TME, which physically blocks drugs from even getting in. You combine that with its natural resistance to chemo, and it's a perfect storm, really. And this right here, this is a brilliant illustration of why it's always diagnosed so late. Just look at this timeline. From the very first mutation to the birth of an actual cancer cell can take about a decade. A decade! Then it can take another 5 years before it even starts to spread. All of this is happening with pretty much no symptoms, which is exactly why we need a way to spot it much, much earlier. So, okay, with a challenge like that laid out, how in the world do you fight back? Well, this brings us to a powerful new toolkit that's changing the whole approach to this disease, proteomics. You know, we hear so much about genomics and DNA, the blueprint of life. But think about it for a second. A blueprint for a house tells you what could be built, right? But it doesn't tell you what's actually happening inside that house right now. Are the lights on? Is the water running? To know that, you've got to look at something else.
And that something else? It's proteins. This is really the heart of proteomics. It's the study of all the proteins, which are the real-time machinery of our cells. They're the workers, the messengers, the tiny little engines. They are what actually carry out the instructions that come from our genes. This slide puts it all side by side, which is super helpful. Genomics, the study of our genes, that's the blueprint. It shows potential. But proteomics, that shows us the functional machinery in action. It gives us a snapshot of what is happening in a cell right this second. For a dynamic disease like cancer, getting that real-time information is absolutely critical.
So how do scientists actually use this new toolkit? Well, they basically become spies, using it to eavesdrop on how cancer cells talk to each other and get things done. Okay, so this diagram here, think of it as the whole game plan. On the left, you've got clinical proteomics. That's looking at samples from patients. But the real deep dive, the investigation, that's happening over on the right in functional proteomics. This is where scientists zoom in on the cellular level. They've found the cancer's playbook, and for the first time ever, they have the tools to actually read it. And if we zoom in on the cancer cell itself, proteomics lets us see these tiny little protein switches getting flipped on or off. Now, the fancy term for these is post-translational modifications, or PTMs. But really, just think of them like the wiring inside a control panel. They're what dictates everything the cell does, from growing and dividing to resisting the drugs we throw at it.
But it gets even cooler than that. We can also listen in on the conversations between the cancer cell and its neighbors, the stroma cells. The cancer cell sends out these protein signals, shown here as little secreted proteins and exosomes, essentially shouting orders at those stroma cells, telling them, hey, build that dense protective fortress we talked about earlier. Okay, so understanding the playbook, that's step one. Step two is using that intel to launch a counterattack and find the cancer's weak spots. So here's the absolute crux of it all. If these cancer cells are constantly sending out these secret protein messages, what if we could intercept them? I mean, the ultimate goal here is to find those signals just floating around in a patient's blood. That would be a total game changer. A simple blood test for early detection. And the research is pointing to exactly how this could work. Take a look at this quote. It's talking about these tiny little messenger packages called exosomes. Cancer cells are constantly shedding these exosomes, which are packed full of their unique proteins. And this research suggests they could be the perfect markers for a reliable test. In this diagram, it shows you the strategy in action. It's actually pretty straightforward. You start with a simple blood sample. From there, you pull out these tiny exosomes, and then Using the tools of proteomics, you analyze the proteins inside them, hunting for that one-of-a-kind signature that just screams cancer. And guess what? They're finding it. This chart here is a super simple look at a key discovery, a protein called GPC1. Look at the difference. It's sky-high in the exosomes from pancreatic cancer patients, but it's barely there in healthy folks. A signal that clean, that's exactly what you're looking for in a biomarker. It's huge.
But this research isn't just about finding the cancer earlier. It's about building a whole new future for how we actually treat it. This work really lays out a clear roadmap. It starts with step 1, discovering these unique protein signatures. That naturally leads to step 2, building a super sensitive blood test. From there, we can get to step 3, creating targeted drugs that go in and block those very specific cancer signals. And the endgame? Step 4, truly personalized treatments based on a patient's own unique proteome. For the longest time, pancreatic cancer has just been this black box. But now, by studying its actual functional machinery, the proteins, we're finally starting to peek inside. This shift, from just looking at the blueprint to watching the real-time action, man, it opens up a whole new world. And it leaves us with this really hopeful question. Are we on the verge of using the secrets hidden in our proteins to finally rewrite the story of this devastating disease?